Hello and welcome to our webinar this evening, The Gut Microbiome and Celiac Disease and Food Allergies. My name is Christy Lansdowne and I'm the Executive Director of Achieving Cures Together. For those that are new to ACT, we are a Minnesota-based nonprofit organization that partners with the University of Minnesota's Microbiota Therapeutics program and researchers around the country like Dr. Weingarden, who's here with us tonight, to study the connection between the gut microbiome and our health. Our partners at the University of Minnesota take microbes from highly screened stool donors and use pharmaceutical grade standards to produce microbiota therapies that are used in clinical trials targeting a variety of health conditions throughout the country. You can see some of our partners here on the map. Our partnership has supplied over 65,000 microbiota transplant therapies to patients suffering from antibiotic-resistant C. difficile infections and clinical trials around the country. Today, ACT supports microbiota research targeting cardiovascular health, antibiotic resistance, cancer recovery, digestive system disorders, the gut-brain connection, and immune system disorders. Our mission and work would not be possible without both the collaborations with all of our research partners, as well as our generous support of our donors. So we want to give a big thank you tonight. We do have a number of ways to get involved and learn more about the research that ACT supports. We do host a variety of webinars throughout the year, like the one this evening, focused on a variety of different health conditions. And we also have an annual in-person event, Growing Hope, which will be hosted this year on Friday, September 11th at the gorgeous winery at Sovereign Estates. You can see a little glimpse of it on the screen here. We're honored to have our guest speaker, Dr. Richard Bedlack, join us from Duke University at Growing Hope to share his research that is using microbiota therapy to target ALS, amyotrophic lateral sclerosis, or Lou Gehrig's disease, which is a progressive neurodegenerative disease. We also have opportunities to join ACT's charity race team. If you're local to the Twin Cities and want to join us this fall, if you're a runner or a walker, you're welcome to join us as well. And before Dr. Weingarden's presentation tonight, we do have a special guest speaker. Rich Gannon, former NFL quarterback, couldn't join us live tonight, but wanted to share a message on the impact celiac disease has had in his family.
Good evening and welcome. I'm Rich Gannon. Some of you may remember me as a former NFL quarterback. I was fortunate to play 17 years for teams like the Vikings, Chiefs, and even the Raiders. Blessed to play in a number of Pro Bowls, was a league MVP, played in championship games, even a Super Bowl. But what I'm most proud of is the work that my wife Shelley and I have done, working closely with the Center for Celiac Research, first at the University of Maryland in Baltimore and now at Mass General. You see, 28 years ago, my youngest daughter Danielle was diagnosed with celiac disease. And at the time, Shelley and I knew very little about an autoimmune disorder that affects so many Americans. We worked closely with the Center for Celiac Research, the University of Minnesota, where she was diagnosed, Dr. Alicio Fasano, so many local physicians. And we had fundraisers, we had walks. We worked with food manufacturers to improve labeling so that moms and dads could understand what was in the products that their kids were consuming. We've worked closely with restaurants in terms of cross-contamination and just really trying to help those who suffer from this disease to have a better life. And I am delighted that Achieving Cures Together is partnering with the University of Minnesota Microbiota Therapeutic Program and supporting this important research. Now, moving forward, please consider ACT a reliable resource for any gut health concerns and related illnesses for your family and friends. I thank you for your participation tonight and your ongoing interest and support of Achieving Cures Together as they advance their mission, which is saving and enhancing lives by supporting the advancement of microbiome research. Thank you again and enjoy your time this evening.
We want to thank Rich and his family for their dedication to advancing celiac disease research and promoting safe food preparation and packaging for celiac disease patients, which is so critical. As we transition to our main speaker this evening, just want to give a reminder that we will have a Q&A opportunity at the end of the webinar. As we go through the evening, please feel free to submit questions on the Q&A section or on the chat. We also have a couple links that we'll post for everybody on the chat, so feel free to keep an eye on that. So without further ado, it's my pleasure to introduce our keynote speaker tonight, Dr. Alexa Weingarden. Dr. Weingarden graduated from the Medical Scientist Training Program at the University of Minnesota in 2017. She completed her PhD in the Microbiology, Immunology, and Cancer Biology program with Doctors Alexander Koritz and Michael Sadowski, where her work focused on understanding the mechanisms behind fecal microbiota transplantation for recurrent C. difficile infection. After medical school, she went on to residency in internal medicine, followed by a gastroenterology fellowship, both at Stanford University. Dr. Weingarden's current research is focused on microbial and immune interactions in celiac disease and food allergies. Please help me welcome Dr. Weingarden.
Thank you for that introduction, Christy. All right. Again, thank you guys. Thank you everyone for having me on to discuss this topic, and of course everyone who's joining us to learn more about it. So I'm going to tell you about the state of what we know currently about the role of the gut microbiome in celiac disease and food allergies. And then I'll also have the opportunity to talk a little bit about the advances that my lab is making in terms of understanding how the microbiome might be contributing to these diseases. So as you're probably aware, the rates of these diseases, both food allergy and celiac disease, are on the rise. On the left, you can see a graph that's indicating the number of emergency room visits for severe food allergies. And it's pretty clear that compared to 2005, over the next 10 years, so going up to about 2014, there's been a significant increase in the presentation of these severe allergies, and that reflects the increased prevalence in the general population as well. And then if you look on the right, the rates of celiac disease also seem to be increasing. This is a study that looked at different studies of celiac disease to look at how common it was at the time that the study was performed. And you can see that compared to certainly the 80s, but even the 90s and early 2000s, there's been a really big increase in the incidence of this disease, so how frequently this is being diagnosed. So with this, there's obviously a big question of why are these diseases increasing? Unsurprisingly, there's a lot of different factors that go into the development of both of these diseases. Genetics, obviously, things like diet, clearly, medication use, changes in the rates of different infections, and of course, as we're focused on today, the differences in the gut microbiome. Now, something like genetics, although we suspect it's involved in food allergy, and it's certainly a part of celiac disease, the chance that our genetics as a population have changed rapidly over the past couple of decades is probably pretty unlikely. So we're probably more likely seeing changes in some of these other factors, again, particularly the gut microbiome, as reasons why food allergy and celiac disease prevalence are increasing. And understanding how the gut microbiome is involved with these diseases is particularly exciting because, as everyone through ACT is well aware, we might be able to alter the gut microbiome in order to treat these diseases. Okay, so for the talk today, I want to cover, first of all, of course, what are these diseases, and give you a little bit of the background into the immunology side, which I think really helps understand how the gut microbiome affects or could be affecting these diseases. Some data on, is the gut microbiome altered in these diseases? Unsurprisingly, it is, but we'll talk about some of the details with that. And then finally, a quick discussion on whether or not there are advances in microbial therapies that could be used to treat these diseases. Okay, so starting off with the basics, and that involves truly going back to basics and talking about how does our immune system normally respond to food? So typically, when we eat food, we generate a state that's known as tolerance. So food proteins, is kind of the biggest example, they're not normally part of our bodies, right? So our immune system has to figure out how to manage them. And in most cases, this leads to a non-inflammatory, basically a non-response or a tolerogenic response where the immune system basically goes, great, these things are good, they're helping us, we're not going to have a problem with them. We know that this is mediated through a set of a type of T cell, a CD4 positive T cell known as a regulatory T cell, and some other related types of T cells. And these T cells, specifically the ones that can recognize these food proteins, are really important for blocking inflammation in response to them. Unfortunately, we think this is going awry in both food allergy and celiac disease. So instead, we think, again, hard to know for sure with some of our current methods, but we think that instead of that normal regulatory T cell response to food proteins, instead you get two different types of an inflammatory response. In food allergy, this is characterized by the development of this type of T cell, that's so-called Th2 or type 2 helper cells in food allergy that are recognizing food proteins and leading to downstream inflammation. And similarly, in celiac disease, you have a different type of inflammatory T cell known as a Th1 or T helper type 1 cell that seems to be responsible for driving the inflammation that we see in celiac disease in response to the gluten proteins that are causative of celiac disease. So overall, we think there's a big shift in terms of how the immune system is recognizing and responding to food proteins compared to normal in both food allergy and celiac disease. Now, a little bit more about both of these diseases, which I think is going to help to understand some of the data on how the gut microbiome can affect the immune system in these diseases later on in the talk. So to review basically how a food allergy response is generated, essentially, again, you start out by having this abnormal inflammatory T cell that's responding to a food protein, those Th2 cells. Those in turn will go on to trigger the development of a type of B cell that's producing an antibody known as an IgE antibody. And those antibodies, they're also recognizing the food proteins, the food particles. Now, when you eat those proteins and they bind to that IgE antibody that recognizes the protein, those antibodies in turn will bind to receptors on a type of cell known as a mast cell. And when those IgE antibodies are bound to the mast cell, that causes the mast cell to release factors that ultimately lead to the symptoms of a food allergy, which when it's out, in addition to hives or rash, we call that anaphylaxis. So that, again, that includes often having those hives, that itchy rash. People will often have breathing problems as well. And then obviously you can have GI symptoms, vomiting, diarrhea, and then and abdominal pain. In severe cases, you can have what we call anaphylactic shock. That's where your body starts to lose blood pressure. Your blood pressure goes down because you're having such a severe reaction to these food proteins via these immune pathways. Now, in celiac disease, also, this is a disease that's characterized by this abnormal inflammatory or immune response to food, but the pathway is a little bit different for how it happens, what the downstream result is. So again, instead of having a set of regulatory T cells that recognize proteins from gluten, including gluten from wheat, barley, or rye, you instead get the development of these Th1 inflammatory T cells that recognize these gluten proteins. And then those T cells, we think, go on to activate a different type of T cell, what's known as a cytotoxic T cell or a CD8 positive T cell. And those T cells are the ones that start to kind of go crazy in the intestine and lead to inflammation. So in individuals with celiac disease, frequently you'll see that instead of these, the normal, what we call villi, so basically the lining of the intestine, which you're seeing a microscopic image of on the lower right, instead of these long villi that allow for a lot of area to absorb nutrition, those villi go flat, a condition that we call villus atrophy, and that's what you're seeing in the bottom right corner, the bottom right image. And you can understand, or you can kind of easily imagine that when you have less surface area to absorb nutrition, you're going to have problems absorbing things. And that leads to a lot of the malabsorption symptoms that we see in individuals with celiac disease. Okay, so now that we have the basics of what is happening on the immune side in food allergy and celiac disease, now we can talk a little bit about how the gut microbiome is altered in these diseases and how changes in the gut microbiome might be affecting different parts of the immune system that could trigger or in some cases maybe help prevent these diseases. So unsurprisingly, or I probably wouldn't be giving this talk, the intestinal microbiome is different in both patients with food allergy and celiac disease. So what I'm showing you here is just one example of a study that's looked at in this case, the stool microbiome in patients with a food allergy, specifically in infants with a cow's milk allergy, or what's designated as CMA on this figure. So what I'm showing you here are the results of stool DNA testing to look at different microbial groups, what we call taxa, in the stool, again, of individuals with CMA or in healthy infants. And what's pretty clear is you can see that there are a number of different taxa, these groups of bacteria that seem to be higher, more prevalent in infants that had a cow's milk allergy. And in contrast, on the right side, you see a different set of bacteria that seem to be more prevalent, more common in healthy infants. So yeah, pretty clearly there's been a shift in the types of microbes, especially the types of bacteria in individuals with this cow's milk allergy compared to healthy infants. We can find similar things when we look in individuals with celiac disease, again, compared to individuals without celiac disease, or the control at the bottom of this figure. So this is a little bit more complicated. I thought this was very interesting because in addition to looking at the microbial content of the feces, the stool in individuals with celiac disease or control patients, this study also looked at gut microbial populations that were associated with duodenal fluid from the small intestine. That's right next to where that disease is actually happening in the small intestine. And microbial populations that were actually bound to the intestine that could be released when they took biopsies of the duodenum, the small intestine of patients, again, with and without celiac disease. And what was really interesting is that while you did see differences in the bacteria that were present in all of these locations, specifically which bacteria were different varied depending on location. So that suggests that not only do you see differences in the stool microbiome in individuals with celiac disease, but you also see differences that are present in the small intestine itself. And what might be relevant might be more changed or more important for the relationship between the gut microbiome and the host in celiac disease might actually be some of these differences that are found in the small intestine instead of in the stool. Okay, this was another really interesting study that I wanted to highlight because this was a study that looked at whether or not there were differences in the stool microbiome before individuals were actually diagnosed with celiac disease. So this was a longitudinal study where they took stool samples from babies who were at higher risk of having celiac disease based on some genetic testing. And they took samples from these individuals before they were even diagnosed, starting up to 18 months before these children develop celiac disease. And what you can see is that even prior to the development, the formal diagnosis of celiac disease, there's some pretty big distinctions in the types of bacteria that were present in the stool. And what you're seeing, again, don't worry about the individual details, just note that the types of bacteria and how common they were pretty different in individuals that went on to develop celiac disease, that's on the left, compared to individuals who did not develop celiac disease on the right. So this really says that, hey, maybe there is something that the microbiome can really kick off that could drive the development of this disease. It's not just an association where we're not really sure, chicken or egg, which came first, right? In this case, we know the changes in the microbiome were there before that person had celiac disease. Now, a really, really good way to demonstrate this is actually to take these findings, take these differences and look in mouse models in order to understand whether or not these differences can actually drive disease. So I'm going to start off by talking about some of the work that's been done in mouse models of food allergy to determine if differences in the gut microbiome can really drive the development of food allergy. So this is actually from that same study that I showed earlier, looking at differences in the gut microbiome in individuals with cow's milk allergy compared to healthy infants. So this was really clever. What this group did is they took mice that were germ-free, so mice that have no microbes whatsoever, and they inoculated them either with stool from the healthy individuals or stool from the individuals with cow's milk allergy. And as I'll go through, effectively, the cow's milk allergy stool was unable to prevent the development of food allergy in these mice. So what you're looking at on the right is a difference in body temperature in these mice that had been either kept germ-free or given one of these two groups of stool microbes. And looking at what happened to body temperature after these mice were exposed to the allergen, after they'd been made allergic. So just to back up and give some more context, when we're understanding anaphylactic shock, again, that's that low body temperature, kind of the most severe form of anaphylaxis to a food allergen. When we're looking at that in mice, it's really hard to get a mouse's blood pressure. But when the mice have a drop in their blood pressure, they also have a drop in their body temperature, which is a lot easier to measure. So that's what we're looking at here, is body temperature drop as a proxy for the development of anaphylactic shock. So what you can see pretty clearly is mice that were kept germ-free or mice that were given stool from an individual with cow's milk allergy had this drop in body temperature. They got sick. They were having the allergic response, a severe allergic response. But mice that got healthy stool instead actually didn't have a drop in body temperature. They stayed healthy. They did not develop this food allergy anaphylaxis. Okay, there's been a really similar finding with another group as well. So this is a really similar study where individuals with the stool was collected from individuals who either had a food allergy or were healthy. And then these microbial populations were given to germ-free mice. And then same thing on the right, the investigators looked at whether or not the mice developed anaphylactic shock in response to the food allergen by measuring the body temperature in mice that were exposed to the allergen. And it's a little bit fuzzier here, I apologize, but what you can see is that very similar to the last study in mice that were given either stool from a food allergic individual or were kept germ-free, there was a pretty substantial drop in their body temperature. They went into anaphylactic shock. But that response was much less when these mice were given stool from from a healthy individual. So that suggests that there are differences in the microbes that are present in a food allergic individual compared to a healthy individual that could actually be responsible for driving a predisposition towards developing a food allergy. Okay, so how did this happen? Do we know anything about what the effects of these microbial populations were on the different parts of the immune system that drives food allergy? So again, we talked about how one of the important steps of developing a food allergy is getting these B cells that are producing this IgE antibody that recognizes the food. So what you're seeing on the left side on that figure is looking at the concentration of those IgE antibodies in the blood from these mice. So either, again, mice that were kept germ-free, mice that were given healthy stool, or mice that were given stool from a food allergic individual. And this matches really well what the investigators saw in terms of the shock response in these mice, where mice that were germ-free or given food allergic stool had higher levels of this IgE antibody that we know is important for driving anaphylaxis, for driving food allergy. But that level was much lower in mice that were given stool from a healthy individual. And then on the right side, I'm showing you two figures looking at one of the proteins that's released from mast cells when they bind this IgE antibody and release the factors that lead to anaphylactic shock. So it's kind of a way of looking at whether or not these mast cells were doing their job, which in this case, we don't want them to do their job, but were they actually releasing these factors that lead to anaphylactic shock? And again, same thing as with that IgE antibody. So mice that were germ-free or got stool from a food allergic individual, including individuals with cow's milk allergy, they had much higher levels of these mast cell proteins in the blood after getting exposed to the food allergen compared to mice that were given stool from a healthy individual. So again, it seems like in both of these cases that having microbial exposures from someone with a food allergy really predisposed the mice not only to have anaphylaxis, but this was due to an increase in those food recognizing IgE antibodies and an increased mast cell response. But one thing that wasn't really examined in those studies and that we haven't really had the technology to do, or no one has looked at until some recent work by our lab, is whether or not these different microbial populations actually affected the T cells that kick off this whole response. So what I'll be showing you over the next couple of slides is work that we've done trying to understand the relationship between gut microbial communities and these food allergen specific T cells that again are kind of the first step in driving an allergy response. So in this case, what we did is we looked at two sets of mice that were supposedly were called specific pathogen free. So they're not germ free. They've grown up with a mouse microbiome, just like their normal lab mice. We call them specific pathogen free or SPF because we've screened them for a number of different microbes that can cause disease in mice, and they don't have them. We keep them clean. They're not germ free. However, even within these different groups of SPF mice, there are differences in different mouse vendors, essentially, which is what happened here. So I was looking at two sets of mice that came from two different companies that sell the mice, basically. And these mice have different gut microbial communities. In theory, these mice are otherwise genetically identical. But as you can see, we looked at the stool microbiome of these mice. That's what the figure that I'm showing on the right. And what you can see is that mice that came from one vendor, TAC or Taconic, had microbial communities that all clustered together when we looked at the DNA, the stool microbial DNA content or bacterial DNA content. And this clustered separately from mice that came from from Jackson Laboratory, which is another mouse vendor. What I'm showing along the bottom in the heat map is, again, don't focus on the details too much. There's a lot going on there, but all that you need to know is that we were able to pick out individual different species that explained why there were differences in the overall microbial communities from these mice. So this first part of the experiment that we did just showed, yes, these mice do have different microbial communities. Now, what was really interesting is when we made these mice allergic to a food, we found that there were differences in their allergy response. So the figure on the right is a very similar figure to what I showed before. So it's looking at differences in body temperature in these mice when they were exposed to the food allergen. And mice that came from one vendor, this Taconic or TAC compared to Jax, had a much bigger drop in their body temperature. They were having much more severe anaphylactic shock than mice that came from the other vendor that came from Jax. On the left, that's basically just a different way of looking at an anaphylactic response in these mice. So when these mice are given the allergen, they'll have another symptom that we also see in people of a food allergy, which is diarrhea. They'll get diarrhea within an hour after having the food allergen. And you can see that as we give the mice more and more exposures to the allergen, that's what's at the bottom, this gavage, more of the mice get sick. But the mice get sick a lot faster when they come from Taconic. So overall, it seems like this difference in microbial community drove a difference in food allergy severity, which again, this mimicked what had been seen before. However, what we were able to do is actually look at these T cells in the gut that were recognizing the food allergen. And what I want you to notice here is that the number of T cells that we were able to find that could recognize the food allergen in mice from Jackson, the mice that were doing better, a lot more of them were these, what we think of as the normal food response. They were regulatory T cells, or Tregs for short. Now, in contrast, in the taconic mice, again, the mice that were doing worse, a lot more of these allergen-specific T cells became those inflammatory Th2 cells that we think are really important for driving the food allergy response. So again, what we were able to show kind of for the first time is that the differences in microbial community could affect not only the mast cells, not only the B cells, that IgE antibody, but the T cells as well. And then just to really drive home that this was a microbial difference, it wasn't just due to some subtle genetic differences between these mice. We put them into the same cage, so their microbial communities mixed up together. And that's basically what I'm showing on the right side, is that animals that came from, had originally come from Jackson, but got put in the same cage as mice that came from Taconic, took on the bacterial communities of mice that came from Taconic. The Taconic bacteria basically invaded and either kicked out or decreased the microbes that had been present in mice that were from this other vendor from Jackson. This was also associated with a shift in how severe the anaphylactic response was in these mice. So this is again looking at those whether or not animals got this diarrhea, this anaphylactic response to getting a food allergen, to receiving the food allergen. And you can see that the mice from Jackson, those are the open circles that were housed with the mice from Taconic, the more severe, originally had the more severe food allergy response, also got sicker at the same rate. And indeed, we saw the same thing with T cells in the gut, that the food-specific, the allergen-specific T cells in the gut in mice from Jackson that were living in the same cage as mice that came from Taconic took on the characteristics of the T cells in mice that had come from Taconic. So again, this really drives home that this shift in not only the food allergy response, but also the T cell response, the type of T cells that formed in response to the food allergen, really did seem to depend on the types of microbes that were present in the gut. Okay, so again, just to reiterate, we also wanted to look at whether or not there were differences in celiac disease, whether the differences that others had seen previously in individuals with celiac disease in the gut microbiome were also capable of driving diseases. Of driving celiac disease. So again, turning to mouse models is a way to try to look at this. Now, mouse models of celiac disease are not as well developed as those for food allergies. So the way that we look at whether or not a group of microbes could cause disease is a little bit different. So this was another really interesting study that had been done by a different group a few years back, looking at whether or not there were differences in the ability of microbes from patients with celiac disease to break down gluten, which, if you think about it, it's a pretty important question to ask, right? Because if you can break down gluten better, if there's less gluten that's present in the intestine for the body to absorb, for the immune system to respond to, then maybe there won't be as severe a reaction. You can calm down celiac disease in the same way that you do by getting rid of gluten in the diet. You can also have a microbe that could get rid of it. So that's essentially what these investigators looked at. So they took germ-free mice, again, those mice that have never had any microbial community, and gave them microbes either from an individual without celiac disease, the control patients, or an individual with celiac disease. And then again, they looked at the amount of gluten that was present in the small intestine of these mice after they'd gotten one of these two microbial communities. And you can see pretty clearly that mice who received stool or microbial communities from someone with celiac disease had a lot higher gluten in the small intestine. Really, those bacterial communities were just not as good at breaking down gluten compared to control individuals. Okay. There's also been some work, also by the same group, looking at whether or not different types of bacteria might be able to affect some of the non-gluten components that could contribute to celiac disease inflammation. So this group looked at specifically a component of wheat known as amylase trypsin inhibitors, or ATI. So this is not gluten. It's not what's being directly responded to by the immune system, but when they gave this ATI plus gluten to mice in a celiac model, so this is from a celiac disease model, they found that that villus atrophy, that flattening of the small intestine that leads to malabsorption, was significantly worse than if they gave mice gluten alone or this ATI alone. So this kind of had an additive effect. Now, the next thing that these individuals did, these investigators did, was they tried to find different bacteria that could break down this non-gluten component, this ATI component of wheat. And what they found a type of bacteria called lactobacillus that could break down this ATI. And they gave mice in the celiac model, they gave them the gluten plus that ATI, what caused the lowest villi, the most severe disease. Or they gave them gluten plus this ATI, the amylase trypsin inhibitors. And they gave them bacteria that were not as good at degrading these ATIs. And then they had a separate group of mice that they gave gluten, ATI, and bacteria, these lactobacilli, that seemed to be really good at breaking down ATI. And what you can see is that when they gave these mice those ATI degrading bacteria, they actually saw some recovery. So they saw that instead of having really flat villi, the most severe disease, the most severe malabsorption, they actually started going back towards normal, those long villi that increase the surface area that allow for nutritional absorption. So this suggests that there are bacteria that not only might affect the gluten proteins itself, but maybe they could break down some of these other components in wheat that worsen celiac disease. Okay, some of the work that our lab has done, again, is trying to get at whether or not gut microbes can affect those gluten-specific T cells that, again, are kind of the first step in driving celiac disease. So to do this, we take advantage of a special system that we have here pretty uniquely at the University of Minnesota, known as the Natural Microbial Exposure System, or NME. This is, it's surprisingly simple, but it's a profound concept, which is that you, when you're looking at mice in this specific pathogen-free or SPF condition, they're not really normal per se. We like it because it means that the mice are healthy, which is certainly important, and because everything is very controlled. We can keep things the same from experiment to experiment. But what some researchers at the University of Minnesota discovered was that it's probably not normal per se compared to what happens in humans, right? And they discovered this because if they took mice from a pet store and put them in the same cage as lab mice, those lab mice took on the bacteria that were present in those pet store mice. And they also developed immune systems that now started to look more like human immune systems rather than these SPF mouse immune systems. So I wanted to explore whether or not these natural microbial exposures also changed something about the gluten-specific T cells in these mice. So to do this, I took a set of mice that I kept under these SPF conditions, or a set of mice that got put in the same cage as these pet store mice to give them these natural microbial exposures. And then I looked at the gluten-specific T cells in the gut of these mice when I came back several weeks later. And what you can see, this is pretty early data, but pretty clearly the number of the not just gluten-specific T cells, but those what we think of as the normal healthy response to food proteins, those regulatory T cells, was much lower in mice that were put under these natural microbial conditions compared to SPF mice, which in turn maybe explains why it's been so hard in part to get celiac disease models in mice, because we're not looking under the right microbial conditions. Okay, and then finally, just to talk about some work that we're doing that's ongoing with trying to understand the relationship between these gluten-specific T cells or gliadin-specific T cells in individuals with celiac disease and their gut microbes. So we have an ongoing study where we're collecting blood and duodenal samples from individuals with or without celiac disease in order to look at these gliadin-specific T cell. Gliadin is a component of gluten that's recognized by these T cells. And at the same time, we're collecting both stool samples and those duodenal fluid samples to try to look at differences in the intestinal microbiome of these patients. In particular, we're really interested in trying to understand whether or not there are differences not only in any old microbe that we can find in the gut, but whether or not there's differences in what we call the active microbes. So these are microbes that are actually probably the real, truly resident microbes in the intestine. They're not contaminating from somewhere else. And how we measure this, how we determine whether or not a microbe is active, is by looking at whether or not it can make fresh protein, basically. And to do this, we take, again, that duodenal fluid, the intestinal fluid from patients during an endoscopy procedure, and we incubate it with this reagent called AHA, which gets taken up by the bugs that are doing making fresh protein. They're undergoing translation. And then we can come back in and stain those AHA molecules and with a fluorescent probe, basically. And then we can use flow cytometry to separate out the stained or unstained bacterial components. So we can look at what took up the stain, what was active, and what didn't, the inactive or negative proportions. And then when you have separated those cells using flow cytometry, you can use DNA sequencing in order to look at differences in these microbial populations. And then in this case, not only are we looking at the negative or positive, the active and inactive, but I'm also comparing between celiac individuals and individuals without celiac disease. Again, these are pretty preliminary results, but up front, it does appear that there are differences in the active microbial populations in the small intestine of patients with and without celiac disease. Again, you don't need to get too caught up on the details of which bacteria are increased and which are lower. Just know that on a gross level, it seems pretty clear that the types of bacteria that are active in patients with celiac disease seems to be different than those that are active in patients without celiac. And what's really interesting is that when we looked at individuals who were on a gluten-free diet, they also had many of the same differences in their microbial populations as individuals with active celiac disease. So that suggests these are differences that persist even when patients are doing well, right? Once the inflammation has stopped, there's still something different about their gut microbial communities. I do want to mention as well that these data were analyzed with very grateful for the help from Andrew Sommer, who is a postdoc in the Koritz lab. So many thanks to him for his work on getting me these data so I could share them with you today. Okay, and then just to finish up with some of the work from our lab, these are data trying to looking at those gluten-specific T cells in patients, both from the blood and, as I'll show later, in the small intestine as well. So these are samples. So now we're not looking at bacteria, we're looking at the T cells from an individual with celiac disease, that's on the top, and then an individual without celiac disease who also doesn't have the genes that allow for celiac disease. And then with help from individuals in Mark Jenkins' lab, we've developed a set of reagents to try to find these specific T cells, these T cells that recognize gluten proteins. And what you can see in the second from the left column on the top, from an individual who has celiac disease, we can find these proteins. We can, if we use this reagent, we can detect cells that are binding to it that represent T cells that recognize these gluten proteins. But on the bottom, in an individual without the right genetics, we can't find it. So that's kind of a check that this reagent is actually detecting what we think it's detecting. Now, what's also really interesting is when we look at what's happening with the phenotype, the behavior of these gluten-specific T cells in this individual with celiac disease. So first of all, those T cells do not become regulatory T cells. They don't express the critical marker for regulatory T cells. That's FOXP3. And that's in comparison to other cells that aren't recognizing gluten from that same patient, where we can actually find some of those regulatory T cells that express that FOXP3 protein. And then if you look at the cells that don't express FOXP3, the non-Treg population, in the gluten-recognizing T cells, all of them express a key marker for these Th1 inflammatory cells, what we call CXCR3. Certainly we can find expression of that marker in the non-gluten recognizing T cells from this individual, but there's also a population that don't express it as opposed to these gluten-specific cells. We also found there was a very small population of these gluten-specific cells that expressed a protein that's associated with a different type of T helper cell. That's that CCR6 that you see at the bottom. And then we also we can find these cells in individuals' intestine. So this is just a sample, a single patient, where we looked at the blood. That's the PBMC on the top left. Again, we can find, they're rare, but we can find those cells from this individual. And then we can actually find more cells in the duodenum, the intestinal sample from this patient. And what's really interesting is it looks like, in contrast to what's happening in the blood, some of these cells do express FOXP3. They do seem to be regulatory T cells. So one of the interests in the lab is trying to understand, are those T cells, or regulatory T cells, the Treg cells, behaving normally, or something different happening with them that doesn't allow those patients to have that tolerogenic, the normal response to gluten that we expect to find. And then ongoing work again in the lab is trying to understand whether or not these differences in gluten-specific T cells that we're starting to find, are those correlated to some of the differences that we're seeing in the microbial populations in the intestine. So that's some future exciting work that once we get a little bit more data on both sides, we're hoping to try to understand better. Okay, and then just the final portion of the talk, I do briefly want to talk about what are some of the ongoing efforts for microbial therapies that could be used to treat these diseases. So what are some of the ways that we might be able to treat individuals with food allergy and celiac disease that rely on a microbial therapy? So one that I've kind of hinted at before is maybe we can get microbes that can degrade these problematic food proteins. So it's just effectively the same as trying to avoid eating the protein, right? If we just get rid of it, maybe we solve the problem that way. Another strategy might be to provide a microbial metabolite that's providing some immune benefit to the host that might counteract the severe inflammatory response to these food proteins in food allergy and celiac disease. And then finally, there is some ongoing work looking at fecal microbiota transplantation or microbial transplant therapy for individuals as a way to maybe get back to a normal microbial population and therefore ameliorate these diseases. Okay, so this is some work. It's actually the same group again that looked at whether or not they could give a peanut degrading microbe to mice with a peanut allergy and whether or not that would affect their anaphylactic response to the peanut. And they found yes. So if they gave these peanut degrading microbes, they saw that there were lower levels of these peanut proteins, the food allergen in the blood. That's what they mean by systemic access. And they also found that these mice had less anaphylaxis, so less of that shock response, and evidence that there was less mast cell degranulation, so these mice were getting less sick. There's been some work as well trying to understand whether or not in celiac disease, giving mice this tryptophan, which is one of the amino acids, might actually be beneficial. And specifically the ways in which we think it's beneficial is tryptophan gets metabolized by gut microbes into a variety of metabolites that might be able to improve gut permeability and therefore prevent inflammation. So that's what I'm showing from their data on the right, where when they gave tryptophan, a high tryptophan diet, to mice with the celiac disease, like in the celiac disease like model, those mice had lower gut permeability compared to mice that were given a low tryptophan diet. And then similarly, they were also able to show that these mice in the celiac disease model that were given the high tryptophan diet had lower numbers of inflammatory cells, those CD8 cytotoxic T cells in the intestine, compared to mice that were on a low tryptophan diet. So maybe giving tryptophan or giving one of these metabolites of tryptophan might be beneficial for individuals with celiac disease. And then finally, yes, I would be remiss if I didn't mention that there is an ongoing phase 2 trial of microbial transplant therapy for children with food allergy. And this is actually in conjunction with Alex Koritz with his product with a group in Boston that is giving MTT to children with food allergy to see whether it's safe and efficacious at preventing food allergy. And specifically what they're interested in looking at is whether or not MTT can basically increase the amount of the food allergen that kids can tolerate before they develop anaphylaxis. They had some good data from their phase 1 trial, so they're moving forward into this phase 2 and expanded trial. I haven't seen data yet from it, but I'm hopeful, crossing my fingers, that will wind up being a useful therapy for food allergy. Okay, so thanks for sticking with me, and hopefully you guys learned something today about the role of the gut microbiome as it affects the host immune system in food allergy and celiac disease. I do just want to thank members of my lab, definitely members of the Koritz lab, who've been very beneficial, especially for the work on that we're doing on celiac disease, and then my collaborators across the University of Minnesota Center for Immunology, as well as my funding sources. So thank you all for listening. I'm happy to take any questions. And yeah, I'll just...
Great. Thank you so much, Dr. Weingarden. We're so grateful for you sharing your insights on the complex relationship between the microbiome, celiac disease, and food allergies. We do have a few questions that have come through, and we'll give it a couple more, just another minute for people to continue submitting questions. We do have our one of ACT's board members who is a patient and here to share his experience as a patient in celiac disease research and treatment. So we'll have Brendan Halleron share with us, and then we'll move to our Q&A session. So if you have some more questions, please send them. We will have a good chunk of time for that as well tonight. So thanks so much, Dr. Weingarten, and we'll be back to you momentarily here. And thanks again, Brendan, for joining us as well.
No, thank you for having me, Christy, and thank you, Dr. Weingarten. A lot of info. Hope to understand more of it over time, but really insightful. And so before we get to the Q&A, I just wanted to share kind of my experience, specifically with Dr. Weingarten and this research. And my story with celiac is actually a little bit unique as it came to me later in life. My father has it, and I assume someday, I would develop it as well. And how that came about was a little bit of a wild story. After a trip to urgent care for a variety of symptoms, I actually was originally misdiagnosed as leukemia. Talk about a change in perspective. So you can imagine how relieved I was eventually to be diagnosed with celiac, and soon after found ACT. Through Peter Westerhouse, in the hope that someday there would be someone out there looking for a way to mitigate this condition. Only took a couple of years, but sometime later I was connected with Dr. Weingarten at the ACT annual gala, and after discussion, agreed to be part of her research. So our schedule is incredibly busy, and it's only busy if you're the best. Dr. Weingarten was so thorough in her evaluation of me and getting prepared to be part of this study, and even found some ancillary conditions that I should look into, really unrelated from her focus. It's this attention to detail that's a testament to the level of care she shows all of her patients, and you can see it clearly in the statistics as well. When it came time for the procedure, visited me beforehand to check in, answer any questions, and just really explain the process and what was going to be happening to me and how I was going to help her research. And it was this level of care and compassion, I think, during the entire process that really should be the standard for the rest of how medical professionals should be adhering to. She's the perfect example of what this organization is for and who it exists to serve, people making a difference. And that's really the whole point of ACT, not only raising money, but helping to create a community of connections to advance this research. So I'm really grateful to have Dr. Weingarten, looking after me, and would gladly be patient number 19 any day to support her desire to change the lives for others.
Thanks so much, Brendan. Oh, each patient story is so valuable, and we're grateful for the detailed level of personalized care you received. Dr. Weingarten, we're grateful for you and your team and all of the research that you do, and we're hopeful that expanded research will continue to pave the way for improved patient care and treatment. And we're excited to move forward now into our Q&A. We do have some questions lined up, and again, feel free to submit more as we continue if you have a question that comes up. So I'll start with our first question here. More of a general question about FMT treatment. When will people who have serious GI issues and need of an FMT be able to get this treatment? Are there options or opportunities for doctors or hospitals for individual for FMT? They're also asking with FDA regulation for C. difficile and ulcerative colitis clinical trials, those opportunities are few and far between and hard to qualify for. So generally just wondering, what options are there for patients who are experiencing symptoms and really interested in FMT, and how does that process work?
Yeah. Your question raises a really critical point, which is that with some of the changes in the FDA regulation of FMT, or microbial transplant therapy, as we sometimes will refer to it, it's been very challenging for patients, even those with C. difficile, to receive appropriate treatment for their condition, which has been very challenging. I do know that Dr. Koretz is associated with a number of studies really across the country for ongoing trials of MTT in a variety of different conditions, both inside in the gut and outside the gut. And I would definitely refer to ACT's website and to the MTP, the Microbial Transplant Therapeutics Program, at the University of Minnesota's website for more information about studies that are ongoing that could be applicable. That's probably the best way to try to understand what's kind of the cutting edge and what trials might be out there already.
Thank you so much. Yeah, there is a lot of great research going on and so many advancements made, but we know it's hard for patients who are in the thick of symptoms right now. And we'll do our best to connect patients with any active clinical trials that they may be eligible for. And really appreciate you sharing kind of that journey and that process for FDA regulation. We do have to wait on that sometimes, but there is a lot of great research happening. So thank you so much, Dr. Weingarten. The next question, have you ever thought of using omalizumab to control the mast cells?
Yeah, no, that's a very smart question. It's so smart that there was a recent New England Journal of Medicine study on it. So you're thinking along the right lines. That there is that study that came out within the past couple of years, looking at using omalizumab, which is an anti-IgE antibody. So it's an antibody that targets an antibody, if that makes sense. It's blocking those problematic IgE antibodies. As a therapy for food allergy. Omalizumab has been a treatment that's been available for a while for asthma, but it was very exciting to see it be repurposed successfully for food allergy. So yeah, I think that's, there's a lot of different ways that you can think about new treatments for food allergy, as well as celiac disease, any of these diseases. One is microbial therapies, which is a lot of what I focused on here, but absolutely there's a lot, there's a reason I studied the immune system too, right? There's a lot of really important insights that we learn from studying the immune system of these diseases that do lead to new therapies. So yeah, really good question.
Definitely. Yeah, thank you. One question that kind of leads off of that slightly here. Do you see any connection or possible application of the advancements being made through microbiota therapy research in food allergies to seasonal allergies or even asthma?
Yeah, that's a really good question, a really pertinent question too. So there's actually been work from what's her name? I'm so sorry. There's several different groups looking at differences in allergic airway disease like asthma in individuals living in either more rural communities or in like Amish or Mennonite communities where they have greater exposure to farm animals, for example. And those individuals do have much lower rates of allergic airway disease. So that suggests that, yeah, there is something also happening involving environmental exposures, probably microbial exposures, and asthma. And I would not be surprised to learn that there's work also being done for things like atopic dermatitis. So another allergy and asthma related disease. Yeah, good question. There's probably connections there. Yeah, absolutely.
Great, thank you. Another question we have is any suggestions on ways we can proactively strengthen our microbiome to prevent food allergies?
Yeah, really good question. So there are there's some work, again, it's connected to some of the studies that I've shown, looking at what microbes or microbial components could be involved in protection against food allergy. One of them is a group of bacteria known as Clostridia. So not C. difficile, different now differently named, but that make some metabolites, notably short chain fatty acids, that might be protective, probably by increasing the strength of the gut barrier. And may also have a protein component. So a flagellum is what it is. It's a part of the bacteria that basically it's like a little motor, allows them to move around places. Both of those seem to be protective for food allergy. There was also a study from, I want to say 3 or 4 years ago, that looked at giving, again, this was just in mice, but giving mice inulin, which is a type of fiber, non-digestible fiber, and that seemed to promote changes in the gut microbiome that led to protection against food allergy. Now, I'm not saying, go out and eat a ton of fiber and that'll protect you against food allergy. Just in the future, that might be one way that we could manipulate the gut microbiome to drive a beneficial effect in individuals with food allergy.
Great, thank you so much. We've got a couple more questions here. So I'm scrolling back over. One question here is, what about tryptophan for healing the intestinal barrier?
Yeah, really interesting. So that is kind of something that was sort of hinted at in that celiac disease study in mice that I talked about. That same group has gone on to give is starting to do trials in human patients as well. So I saw an article from them a couple of years ago where they were treating healthy individuals, so not yet people with celiac disease, but healthy individuals with extra tryptophan, high tryptophan, basically. And they saw some of the changes that they were seeing in mice. In particular, a lot of those beneficial metabolites that they think are made from microbial breakdown of tryptophan, they saw them increased in the blood of patients who were given the high tryptophan. So that might be, I think that group is thinking pretty strongly about that exact question. So it's a good question. It's on the right track.
Great. Another follow-up question is, could you use Xeljanz to control JAK pathways?
Yeah. So this is another immune modulating pathway, a relatively new, relatively new. It's not that new anymore, but a different immune controlling pathway. There is some work that's being funded currently by the Food Allergy Fund to look at not Xeljanz, but a related molecule called abrocitinib, I believe, that, like Xeljanz, blocks those JAK kinases, blocks the JAK protein. So yep, people are definitely thinking along the lines of potentially using medications like Xeljanz to treat food allergy.
Great. Additionally, have you found any connection between the microbiome and EOE?
Yeah. So there was a study a few years ago that looked at the esophageal microbiome of individuals with EOE. So EOE is eosinophilic esophagitis. It's a condition it's basically a food allergy that's in the esophagus. You see a lot of inflammation, and it does seem to be responsive to particular types of foods, many of which are also common food allergens in the more traditional sense. And this, so to go back to the study, they basically took scrapings of the esophagus in individuals with EOE, and they had lower abundance of actually those lactobacilli bacteria that seem to be beneficial, maybe in some of these mouse models for other conditions. What's really interesting is that the type of bacterial community in the esophagus is probably pretty different. I mean, it's different all along the GI tract, but it's probably quite different from what's in the small intestine, and certainly different from what's in the stool. And part of that is because the lining of the esophagus is different than the lining in the intestine or the colon. And it's probably more permissive for those lactobacilli bacteria. So it's actually been very interesting to think about, hey, this could be these lactobacilli as a probiotic, kind of, maybe thinking about an upcoming question, have been of great interest, but not great success. But in some place like EOE, where that's actually an environment where they're found in people and they like to live, they can bind to that surface in people, that might actually be maybe someday a feasible option as a treatment for EOE. That's many years down the road, but it's an interesting thing we're thinking about.
Great. Thank you so much for that explanation. I think you've already maybe set the ball for our next question, but I don't know if you have any other things to add about recommending any particular probiotics for known dysbiosis.
Yeah. So just despite the potential promise of some of these things, no. Actually, with at least for C. difficile infection, there was a study in 2018 that looked at whether or not treatment with a probiotic in people who had been given an antibiotic who were at higher risk of C. difficile, whether or not that did anything to the gut microbiome, and it actually made it worse. So it actually made it take longer for people to go back to kind of a normal microbial community, stool microbial community, after receiving the probiotic, compared to people who got antibiotics but didn't get the probiotic. I mean, there are few cases where we know that probiotics are truly harmful, but there's not a lot of clarity on whether they help. And again, there's sort of a hint that it's specifically for that question, right? For dysbiosis, they may actually be harmful rather than beneficial.
That's helpful to know. I know that can be disappointing when, it's easy to want, something that can help solve the problem, but I'm really glad that you shared that. And yeah, thank you for answering that question. Next question is, does maternal immune activation set up the microbiome to mold itself to allergic form?
Yeah. Interesting question. I don't know if I can quite answer exactly that. What I can say is that there is, at least in mice, there is a pretty clear immune activation process that happens at the time of weaning. So going from just consuming mom's milk, right, to eating solid food. Probably something similar that happens in humans, but a little bit harder to study. But in mice, if you don't have a gut microbiome, so in germ-free mice or mice that are treated with antibiotics, that really disrupts that process and predisposes, we think predisposes those, at least the mice, to allergies and other diseases like inflammatory bowel disease later in the life of that animal. So yeah, there is something really important for kind of how early life microbial exposures, especially at the time of weaning, can really instruct the immune system, how that develops later in life.
Thank you. And I know you shared there are some current ongoing clinical trials, particularly for celiac disease and also food allergies. Do you have any information about how interested patients could potentially learn more about joining study?
Yeah, so I mentioned that MTT trial for food allergy that's ongoing in Boston, but obviously Dr. Koritz and the MTP, the Microbial Therapeutics Program here at the University of Minnesota is connected to that project. So I would bet that somewhere on the MTP website you could probably find information about how to get in contact with the team to see if getting involved would be feasible.
Great. Thank you. Another question, how does histamine influence the inflammation, and could taking DAO help?
Yeah. Okay, so histamine is, we think, a really important part of the final steps of the food allergic response. It's probably not the only factor that's released by mast cells that drives the anaphylaxis, but it's definitely an important one. And then DAO is diamine oxidase, which is a enzyme that breaks down histamine. But to the best of my knowledge, that doesn't seem to be in use as a therapy for food allergy. And it's possible, though I don't know much of the data around that enzyme. It is possible that it may just be too late, right? Like by the time you think to take that medication, it may be too late to have an effect on the histamine that's already been released. I don't know. There's possibly many other explanations, but although we do have medications that do target histamine that are very successful for food allergy and other forms of histamine driven diseases, I'm not aware that DAO is a major enzyme that's used for that purpose, even though it seems like it should be.
Great. Well, thank you so much for lending your expertise to us this evening. We're just so grateful that you could join us and share your knowledge with us and the incredible research that you're doing. We just want to, thank everybody all for joining us tonight for the webinar as well. If you have any additional questions, you can reach out to us at info@achievingcures.com. If it's a question for one of our researchers, we'll do our best to get you answers or connected to a medical provider, as well as we will be sending out a copy of this webinar recording. And so if there's someone that you know that's struggling with celiac disease or food allergies and may be interested in watching, feel free to share the link. Just want to thank everybody again and hope you have a great evening.